Hello, and welcome back to Glycan Hub, the podcast in which we explore health, disease, and longevity through the lens of glycobiology. My name is Rina, and I am your host. In this episode, we will explore the role of glycans in normal physiological function and the consequences of glycosylation defects. As you might already know, glycosylation is a vital biological process that involves attaching sugar molecules, called glycans, to primarily proteins and lipids, but also other molecules. What you might not know is that every single cell on Earth performs glycosylation, and these glycans significantly influence the structure, function, stability, and localization of their target molecules. In earlier episodes, we have already explored their crucial roles in communication between cells and the regulation of the immune system. But my guest today will introduce us to the consequences of genetic mutations which disrupt the glycosylation pathway through his work on Congenital Disorders of Glycosylation, or CDG for short. We'll discuss how different types of mutations impact the severity of the disease and the challenges and limitations of the current treatment options. Listen on to find out why he believes that mom power is the strongest force in the universe, as well as how his chance discovery at age 20 contributed to the development of PCR, a technique that revolutionized molecular biology, enabling groundbreaking research and medical advancements. My guest today is the director of the Sanford Children's Health Research Center and the Human Genetics Program at Sanford Burnham Prebys Medical Discovery Institute. His research focuses on congenital disorders of glycosylation, and he is highly committed to searching for novel therapeutic options for patients with these conditions. A warm welcome to Hudson Freeze. Hello and welcome to our podcast.
Thank you.
So before we start our conversation, I thought a good place to begin would be to ask you what your road to the field of glycobiology looked like, and particularly what is it that still keeps you interested in this field?
Well, I think the thing that led me into it was my first introduction to it in my thesis work back in the early to mid-70s. And I thought, this is something that nobody else is working on. Maybe there is going to be an opportunity for somebody who goes a little off the beaten path to do some things. And I think that I started there. Some people came to me for analyses, and the analyses were about cells that couldn't do glycosylation correctly. And I thought, well, that's interesting. The other thing is that I have a disabled sister. And so when I heard that there were opportunities to actually work in a field that might have something to do with helping people with disabilities, I thought, well, this, my prior life, my earlier life got me ready for this. So, and the rewards that come from the patients in working with them, that's, you can't beat that.
And as our listeners will soon hear, is that you've continued through your research to keep a close contact with patients and their families.
Yes.
Maybe the best place to make a clear definition at the beginning is just to define the difference between glycosylation and glycation. This is something we get asked very often, so I think it's maybe a good thing to distinguish at the beginning.
Well, I think you've hit a very important point. In fact, there was an article, a prominent article in Science here just a couple of months ago, and it talks about all of the work that was done and looking for genes that might be important, as it turns out, in aging in males and females and different kinds of mice. And one of the genes they hit upon was a thing called DDOST. Well, that's a glycosylation enzyme. But the article later goes on to describe glycation. So they got it mixed up. So glycosylation is the cellular metabolic process of adding sugar chains onto proteins or lipids. Glycation is strictly a chemical reaction where the different activated groups interact with each other and produce these things that in the long run are not good for you. And the advanced glycation end products are what happens when you have too much sugar, and that results in the complication of diabetes. So they're very, very separate. But scientists physicians. Dr. Oz writes in his book about the same sort of thing and mixes the two up. So it's very common. Let's get it right.
Okay, so just for our audience to be sure, we are talking about glycosylation today. And could you perhaps, you just defined what it is, but could you explain how genetic mutations might affect it?
Glycosylation is a very complicated pathway, and there's a whole series of pathways that require substrates, enzymes, environmental conditions within the cell to make it work. So if any of those are disturbed, any of them are disrupted, or mutations occur in any of those steps, you can have insufficient or incomplete glycosylation. And when that happens, there are a myriad of problems that happen in patients. And every cell carries out glycosylation. Every cell on Earth has glycosylation. So it's pretty fundamental to life.
And your work has very thoroughly focused on congenital disorders of glycosylation. What are those?
Well, those we define as mutations in any of the genes that affect the normal completion of sugar chains. We call them glycans. It's an easy way to refer to the overall group of molecules. So any mutation that interferes with the ability to complete those chains, put them in the right place, have them be functional, anything that does that, we would define that as a congenital disorder of glycosylation. And in fact, by looking at glycosylation, you can begin to define what those defects are. And we often, of course, by having patients with these, usually they first run into their physician, who more than likely doesn't know very much about glycosylation.
And how common are these disorders?
They're rare. They're rare. I think some of the most common ones, and there's only a few of those, would maybe have a total of a few thousand patients. For the ones that are probably most popular and being worked on now, that one has about 1,000 patients, but there's not familiarity with it in all parts of the world. So I'm sure because it started in Europe, the U.S., and Australia, New Zealand, that other portions of the globe are beginning to discover them. So that number is going to grow. And
while we're still talking about mutations, could you distinguish between inherited and de novo mutations, and particularly how these might impact the severity and disease manifestation?
There are, of course, inherited mutations that come from the individual parent. If it's an autosomal recessive disorder, as most of them are, then that requires one copy of the mutant gene from each of the parents. That's most common. There are a few that are dominant, meaning that only one copy has to be inherited. And some of those have very different phenotypes than the ones that are autosomal recessive. So de novo or spontaneous mutations can occur in the embryo. They can occur in the parent's germ cells, mother or father. And those mutations can then give rise to children that have abnormalities. And in fact, there are a few diseases recognized that are X-linked. So they're expressed primarily in males. Unless it's lethal, then you never see it in males, because it's in females, because they have both two X chromosomes. One is inactivated. But the bottom line is some of those de novo mutations can have very severe consequences. And of course, the parents oftentimes are looking to themselves, did I do something wrong? Is it me? And in many of these cases, of course, if they're de novo mutations, that's not the case. It just developed. It's very unfortunate.
And I'm guessing depending on at which point in the embryonic development these mutations occur, they might not be present in the entire body.
Exactly. And that's what's important. There's a new disorder that we discovered, 4 or 5 years ago. And now it's actually being found in adults who have seizures. And the gene is one that transports UDP galactose from the cytoplasm into the Golgi. And so these adult patients would have seizures. And, they did exome sequence, genome sequencing, the whole body, and you couldn't find anything. And one of the standard surgical procedures, if you're not responding to medication, is to take out a small portion of the brain that's responsible for those seizures, and then look across that section and see if there are mutations that maybe occurred there. And in fact, a certain number of cases, maybe 10% of some of the cases of those kinds of seizures can be caused by a single mutation in that gene that is localized to that part of the brain. You would have never guessed, but that's the sort of the next stage, I guess, of what we can do with genomics and understanding how these things work. So now it turns it back over to the fundamental glycobiologist to say, well, now let's take a look. What do we know about those cells that are affected? What different kinds of glycans? How are they impacted? So it gives a lot of opportunity for, I guess you can say, cross-feeding. Clinical science informs basic science. Basic science informs clinical science. We're all in the ecosystem.
When it comes to, again, genetic mutations, is it usually common that it's a monogenic disease, or is it polygenic, meaning multiple mutations contribute?
That's, again, another interesting question. Most of the ones that we look at are monogenic, and they're easier to track, easier to trace, understand. But there are some where there are contributions from different heterozygous mutations in the same individual that impact different aspects of the same pathway. For instance, there was a paper out a few years back that showed there were individuals who got deep vein thrombosis, and that's forming clots. Well, the only time they would get that is if they were drinking heavily. Well, we know that heavy drinking can impact glycosylation. And it turns out that some of those people had mutations in the gene that's responsible for one of the major causes of a congenital disorder of glycosylation called PMM2. And so I'm heterozygous for that most common mutation, as it turns out. So I have a lot in common with the patients that I work with. So we're kinshipped. But these people, under the stress of extra alcohol and the stress of not having quite enough enzymatic activity, they would now form clots. And the reason was because most of the clotting factors, essentially all of them, are glycosylated. So if you're not sufficiently glycosylating, then you may be deficient in those clotting factors.
And so if it wasn't for that behavior of drinking heavily, they would have never known.
Yeah, that's right. So thank you, folks, for informing the basic science and letting us know.
But as you said, glycosylation happens all over the body, in every cell of our body. And so how might that impact the different symptoms experienced by patients with CDG?
Because it happens in different parts of the body. One of the most common forms, and I guess even the most common symptoms, are intellectual disability, seizures, growth retardation. And so in those cases, of course, we know that glycosylation is really important in the structural formation of the brain, but also in being able to learn, to have normal neurotransmission. And so those are quite common. And I think you can find patients who have other kinds of disorders, because now there are almost 170 different kinds of CDG. So with that large number, you can imagine that there is a whole spectrum of different impacts. And there's one disease, for instance, that we study that doesn't show any intellectual disability. But what it does is to result in what's called a primordial dwarfism, or the little people. And the little people actually have normal intellectual development, but they're short. They have short arms and legs, and rather short fingers. They have a very characteristic facial appearance. So that is caused by a mostly de novo mutation in one copy of one gene that we know when you have two copies, that gives you a very different and much more severe type of CDG. So depending upon the load that the individual type of cells or the organs have to deal with, that will sometimes determine what the outcome is.
And you mentioned, if I'm not mistaken, there's currently 170 known CDGs.
Almost
170. Just so that our audience can appreciate the speed of development in this area, how many CDGs were identified when you first started working in this field?
One. And that was a disorder called eye cell disease. And eye cell disease was considered a lysosomal storage disorder. And the reason was because you accumulated material in the cells that's usually lethal by about 7 or 8 years old. But the reason that you accumulated all of these things within cells was that a molecule called mannose-6-phosphate couldn't be added to a sugar chain. And that was important in targeting lysosomal enzymes, very specifically to the lysosome. And if they don't go to the lysosome, of course, they can't do their work of digesting the material in the cell. And they were secreted. So it was a secretion defect. But in fact, even though it resulted in accumulation of things within cells, that was a secondary effect. The primary effect was not putting on mannose-6-phosphate. So one disorder was known. And I think we've been involved either directly or in collaboration with other labs with the discovery of about 25 or 26 of these disorders. And I was working in iCell at the time it was being worked out. And my sort of original work, believe it or not, in slime molds identified some mannose-6-phosphate there. So it was a natural evolution to go from there to working with these rare disorders and with the patients.
Funny how we have stuff in common with slime molds.
Isn't it?
You mentioned exome sequencing, and now maybe focusing a little bit on research developments in this area. How has exome sequencing contributed to the development in this area? And especially also maybe good to define what exome sequencing is.
There is the whole genome, I think, that people will be familiar with. And that genome actually has about 98% of the DNA that's never made into proteins. That portion that's made into proteins and is sort of thought to be the sort of activity and functional aspects, although the rest of it is very important as well. That's from a series of exomes. So they're introns and exomes. Okay. So the introns are not part of the finished protein, but the exomes, those are made into the proteins. That's only 2% of the genome. So when people look at exome sequencing, they're only looking at 2%, but most, but not all of the diseases come from mutations in that portion. Although the other parts, the introns, will also sometimes have an effect. But exome sequencing has been an absolute breakthrough. I mean, I look at the growth of these disorders from when we first discovered them, and then it was sort of using bootstrap biochemistry to figure out what the genes were, and then went back to show that there were mutations in those genes. Now with exome sequencing, it's reversed. So you have these really wonderful and now fairly inexpensive ways to be able to see different mutations. And with computational biology, you can figure out which mutations are likely, but not guaranteed, are likely to be the cause. Well, with that, you can now go back and look at the glycosylation in the cells or a biomarker that comes from the patients, and you can be able to tell, yes, it's very likely that those mutations are causing this problem. It's been a tremendous breakthrough.
And do you have the information of how many disorders have been discovered or identified since exome sequencing began to be used?
I could probably get, some reasonable idea of this because I have a graph that I actually had put together. And if you say that, let's say the exome was done, I don't know, 2001, 2002, but it was fairly expensive, so not everybody had it. At that point, we knew, about maybe 25 disorders. Now we're up to 165 or more. So without exome sequencing, we'd still be scrounging around and would not have discovered these things.
Something I found very interesting about you when I was reading a bit about you was your contribution to development of PCR. Now, I think most people have probably heard about polymerase chain reaction following COVID. We've all had our tests, but otherwise it's also used in exome sequencing, and your contribution was through the discovery of a microorganism called Thermus aquaticus from the springs in Yellowstone National Park, if I'm not wrong. And so what was it that you were investigating at the time?
Basic science. And again, the solution to medicine is going to be found in basic science. So at the time, I was a 20-year-old student, had never been out of the state of Indiana to speak of, and my mentor, Tom Brock, said, well, look, we're going to Yellowstone this summer. I said, why are you going to do that? He says, well, there are these hot springs in Yellowstone, and we want to see if there are bacteria that can grow there. I said, yeah, that sounds really nice. In part because my high school science project was about life on Mars. So I was accustomed to already thinking about extreme environments, right? And so here was something that was at the other end of the spectrum, from cold to almost boiling hot. So I was there for about 5 weeks in Yellowstone, working with, going out in the field, collecting samples, doing all the basic research, because we had no idea that this would lead anywhere. It was fundamental science. It was funded by the National Science Foundation, who said, well, yeah, it's kind of interesting. Go take a look at that. And so I used as my senior research project the discovery of these organisms, and it was only about a month later I had samples back in the lab, and we incubated some of these from samples from different springs, and we incubated them at about 72 degrees centigrade. And I'd pick up the tubes every day and say, there's nothing growing. One day I picked up the tube and there it was. And I thought, oh my God, could this be something? So I real fast went over and took a sample and put it under the slide. And here are all these bacteria growing. They're all over the place. And talking about it even now, I still get goosebumps. It's...
No, I bet.
Because now I had seen something that nobody had ever seen in the world before. Oh, this was so cool. So at the time, it had no application. And then as time went on, exome sequencing wasn't developed, but the polymerase chain reaction, a key component of that was using an enzyme, the polymerase, that we were able to see in Thermus aquaticus. And surprisingly, we didn't know this at the time, but in about 2013, the U.S. Congress voted to be able to give an award called the Golden Goose Award to discoveries that appeared to have no use whatsoever. It was a waste of money. Why do you put money into this when you could be doing something important? And then discoveries like that then later went on to go, oh my God, you changed the world. Who would have guessed? So anyway, that was a proud moment.
No, I bet that's why I decided to ask you about this. It's crazy. So you went on an expedition, not having any idea if you were going to find something. You found a bacteria. Later on, an enzyme was taken from the bacteria and has been incorporated into a reaction that's now modernized medicine and basic research. It's crazy.
I would say it actually has created the biotech industry. I can guarantee you there's not a company on the face of the earth that's doing biotechnology that doesn't use PCR. So whether this is hundreds of billions or trillions, I don't know. I guess we have to see about the next pandemic.
Coming back to congenital disorders of glycosylation, I'm curious, are animal models used in research and how accurately can they represent human conditions?
Animal models are really essential, and you know, especially if you find a drug, use a drug, you have to test those in animals. So you need an animal model to be able to show that you have efficacy before you can ever take something like this into humans. So that's going at the far end. But if we take it from a research standpoint, we want to be able to make more discoveries. And the best way to do that is to use cellular or organism models of the disorder. And in many cases, those have actually revealed a lot of things that we hadn't expected. So they're absolutely essential. And I know that we have done experiments in mice. Many people also do their experiments in mice. But there's also other systems that are used, and that includes flies like Drosophila, worms like C. elegans, and yeast. So oftentimes it's easiest to work on yeast because they grow fast. You can do the genetics easily. You can find a phenotype. The problem can be that that phenotype is limited to what the yeast is going to do, you know, make bread and beer and grow itself. So you need something that is going to more mimic human physiology. And I'll give another example of something that we did. We made a mouse model of one of the diseases that we discovered. And when we made it, it didn't have any phenotype that we could see. We expected to see certain things going wrong. It didn't happen. It's only when we stressed the animal that now a phenotype appeared. And so that was really important because it said, here is what you need to have a human have a disorder. Maybe you need much, much less before you see it happening in the animal system. So making those transitions can sometimes be difficult, but I can tell you some really important breakthroughs have come from looking at simple model organisms. And we can talk more about specific therapies if you want, but they're absolutely essential.
Before we go into therapies, something I want to talk about is education in the medical system. Now, we said the number of CDGs has increased very rapidly in the last few decades. And so I imagine there has been a pretty big educational gap concerning education of clinicians. Have these discoveries been adequately incorporated into medical syllabi?
No.
No.
In a word. And I think it's important to point this out. Most basic scientists are not intimately familiar with glycosylation. And I think the reason is because people may know, biomedical scientists know that proteins exist, but they're much more focused on proteins and DNA and RNA and things like phosphorylation, where the tools are available to understand this better. You could take all the glycobiologists in the world and put them into a small gymnasium in Indiana watching a high school game, and that would be the entire field. And you look at neuroscience, there's 100,000 people. So the familiarity with glycosylation and glycobiology and glycochemistry is not that common. And if you're training physicians, most of the time basic scientists are participating in that. But if that knowledge isn't there, then the physicians who are being trained are not knowing about it. So there has been a big gap, an immense gap for what physicians will know. You have to be a highly specialized metabolic physician or a geneticist to probably even have heard of this. And I've heard some tragic stories, about physicians who weren't aware of these and could have put patients on therapy that would have made a total difference in their lives. And, you can't ask every doc to be familiar with every disorder, and I get that. But we do need to get out there more. We do need to talk more. We need to integrate these into undergraduate and graduate courses. I just gave a lecture on that, on glycobiology and CDG in the fundamental biomedical course. And there are a couple things that help out. One was when Harrison Ford made the movie called Extraordinary Measures, and he gets to use a line in there. He says, "What's the matter, Sal? Not up in your glycobiology?" So I use that, and I mean, that's a bit of a hook to bring people into, well, what's this guy going to talk about? And then he has another line. He says, mannose-6-phosphate. And again, it works for being able to do the things that we're talking about. That helps. And I think another additional boost is going to come out because Carolyn Bertozzi this year won the Nobel Prize for applying real clever chemistry to glyco, to cells, to organisms. So I think these techniques are now going to elevate the awareness of glycosylation. And Carolyn has been doing this for a long time. She's absolutely brilliant. And she's made so much contribution to the field. So there is a gap, but we're trying to close it.
For our audience who might be composed somewhat of clinicians who are interested in finding ways of perhaps educating themselves more on this, are there any resources you might want to point out?
Sure. There are a couple of family groups. Now, the family support groups have really grown in the last, I would say 10 years. And it is, it's really quite impressive because there is, no force stronger than mom power. And so you have a lot of mothers and fathers, whole families who have organized to get behind, I'm going to take care of my child. And so they form these individual groups. So, one of them is called CDG Care. That's resources for the patient. So if a doctor would happen to find something, he can now take that and say, here, you go look at this. And the doc can get educated as well. Now, there's also a consortium that has been pulled together, an NIH-funded consortium in the U.S. It's called the Frontiers of Congenital Disorders of Glycosylation Consortium. And that is a consortium of about, about 15 different sites. And they're primary clinical sites in the U.S., but we're also expanding those into areas in Europe and beyond. And so the idea there is we're going to collect natural history of different disorders, enroll the patients so that they can have annual checkups or semi-annual checkups. And then we'll have the material because we're also looking at biomarkers to be able to stage these. And I can tell you now that's really pretty amazing. 3 years ago, there were no therapies out there. But now there are about 5 different therapies that are at the point of entering or almost getting ready to enter clinical trials as treatment for some of these disorders. And if you go to the FCDGC, you'll find those resources there. And if you're a physician and you think you want to join this group, you want to become active, you're welcome. And there's going to be a lot of welcoming people, families, clinicians. This is all based at the Mayo Clinic in Rochester, but as I say, there are sites all over the country and expanding worldwide.
Excellent, thank you. And how do families and patients with CDG, how do they get involved in your research? How has that been organized?
Well, in the earlier days, let's say, in the time when there's only 20, 30 disorders, parents, again, mom power, would hear something about glycosylation and they had some evidence of something being wrong. They would go to the internet, they would find me. I still quite frequently get calls and emails from families saying, "Can you help us? Can you just tell us anything?" And then sometimes what I would do is say, "Well, I need to talk to your physician," because that's the primary relationship. And those physicians would then loop me in, and we would get a functioning ecosystem, where we might be able to advise the physician about what to do and the samples that we might need. And so that was our primary connection. Sometimes, of course, it would be the other way around. A physician would be contacting us and say, "I have a patient. What can you do? What resources can you give me? Can we be sure that this is actually a disorder of glycosylation?" And we've done that in many, many instances. And, I think our basic science papers now may be even fewer than our clinically oriented ones, because, I view our job as being the interface, right? Being able to link the communities together. And that's what's important, because, come on, this is about the kids and the adults that have CDG, yeah.
So your institute has organized these patient days every year, where you bring families with patients, you bring clinicians, you bring scientists, and allow them all to interact and meet each other. What has been the result of these meetings?
Outstanding. I know that there are scientists that I've invited this whose research might be peripherally involved in CDG, but they can make a contribution. And so when they come in, sometimes they're a little shy. I've never met a family before. And they're sitting in the midst of the families. They're seeing the kids playing on the floor. And some of them get a little bit choked up. And they'll tell me afterwards, "This is the best meeting I've ever been to. This really changes my life in the lab, because I see what we can do." Sometimes you need a refresher course to do that. And the families benefit because they may get some time with their individual clinician and get to know them as people. And sometimes the physicians get to know the patients in a different way. We have one patient here in San Diego. I just love this guy. His mom describes him as 7 going on 34. And he wasn't diagnosed till a couple of years ago. And he is just charming. And you can tell he's got a disability, but he can speak. He can walk. He has problems, but he can play the drums. And he does a marvelous job of playing drums. I got a video of this kid, playing drums with a blues band. And there's another patient that I absolutely love. She is an equestrian. She does dressage. She's in Australia. And she came to one of these meetings, and we were able to have her hook up and go to the stables of one of her heroes who had been in the Olympics. And he got to take her around his stables. She got to ride on the horse. And then later, because she was part of the meeting, she got to demonstrate that she does dance. She dances, even though she's in a wheelchair. She will be on the ground and she'll make these beautiful motions. And then in the wheelchair, moving around, and she insisted that I sing a song with her. So again, these are the things that link people together, that take us out of where we normally would be and say, we're part of this ecosystem. We're all together. So families love it. Physicians love it. Scientists love it. We do it every couple of years.
Sounds really, really, really wonderful. And Do people from all over the world come to this meeting? Is it an international?
Yes, there are. But I have to say that there is also another international meeting that is coming up in the near future. And that is being organized in Europe. It's called the Congenital Disorders of Glycosylation World Conference. Now, it's primarily centered in Europe, but people from the US and Australia, and I'm sure some of the other areas, such as the Middle East, are also coming. Now, that meeting is going to occur July 21st to July 23rd of this coming year, and that's in Lisbon. And I can send you a link for that so that if people want to get more information, they can look it up on the web.
Excellent. We'll put it in the description for the episode.
All right. Sounds good.
Some of your work is also focusing on the development of new molecular diagnostic methods. How do you aim to improve the current diagnostic options, and what are some of the challenges that need to be overcome?
Well, I think there are some disorders that are still not easy to diagnose. And some of those are because, one, again, they may not be well known, but there are a considerable number of these. These are ones that are called GPI anchor deficiencies. And so there are people now at the Mayo Clinic who are actually working on some new methods using, it's called Codex, which is a way of being able to tag many, many proteins with heavy isotopes, and then by mass spectrometry, look at that. So that is going to be useful. It's very early stages of development, but you can then begin by using just a blood sample to see in those cells what proteins are missing, what proteins are not, and maybe you even begin to sort out different groups with different types of disorders. That's one thing. The other thing that is really quite exciting is that there is the repurposing of a drug called apalostat, which is licensed in Japan, and it's for treating diabetes. And one of the basic scientists, Ethan Perlstein, did a screen looking at approved drugs and asking which one might be able to improve a worm model, C. elegans model, of this major disorder. And he found one, and this was the apalostat. The apalostat turned out to not only improve the biochemical features, but it also, in N=1 patient, you got to start somewhere, improved her condition. So now there are clinical trials that are just starting where patients with this disorder, it's called PMM2-CDG, will be able to take this drug, be monitored for their improvement. And it looks very promising. Now, one of the things that was interesting is that apalostat is an aldose reductase inhibitor. And what it does is to prevent sugars from being turned into sugar alcohols. It turns out that these CDG patients that are part of this clinical trial have elevated levels of sorbitol, which is the reduced form of glucose, and of mannitol. And when you put the patient on this, you can actually reduce the amount of those back to normal levels. So you can take a standard assay that was used for monitoring diabetes, sorbitol, and you can now use it for CDG. And using things like this is a real godsend, because now we can take things we know and apply them. We can take new things that we're developing and apply it. So again, it's the ecosystem. It's the balance between basic science, biomarkers, model organisms, and the patients, with the docs linking us all together.
No, I especially like the idea of being able to repurpose something that was developed for an entirely different purpose into a disease that they probably didn't even know about when they were developing the medication.
No, and nobody would have cared, because I can probably with some confidence say that most people in glycoscience never think about sugar alcohols Well, we should. But there again, coming from something that benefits a patient, you take it back and say, "Oh, we got to rethink glycobiology and how different molecules are being processed within the cell." The stuff that we never really cared about. Maybe that's influencing, and we know it's influencing glycosylation. So there's all this framework of metabolic things going on inside the cell. They're not just a big mixture of things, but maybe they're actually looking more like the Tokyo or the DC subway, where there are all these interconnections and all these pathways. We've never defined them, but they're important.
Now, one treatment option that I found very interesting was the application of dietary supplementation in these conditions. And so where is this type of treatment applicable?
I think it's applicable in a limited number of diseases. The sugar supplements themselves are sugars you can buy them on Amazon. But most of them essentially all of them have not been through clinical trials. So if there is a little bit of literature out there that shows, maybe on even a couple of patients or just even in cells, that there can be improvement, then families will go to Amazon or wherever and do this themselves. We try to encourage people not to do it, because then you can never get the real data that will allow you to then say, this is how much, this is how frequently. And so we first came up with the idea of using mannose as a treatment. And it takes care of most but not all symptoms of a deficiency called MPI-CDG. An MPI-CDG is caused by a deficiency in, believe it or not, making mannose 6-phosphate. And it's converting fructose 6-phosphate to mannose 6-phosphate. But if you provide mannose, then mannose just runs right around that block, says we don't care, and you supplement. So that's important to be able to do that. And you say, well, there's probably no side effects. Well, we found out, again, using a mouse model, that if you give a mom who would be a carrier of this disorder, if you give her mannose, that this in the embryo can result in blindness. And we would have never predicted this, blindness in the embryo. The lens doesn't develop. Now, is that going to happen in humans? Well, we don't have an idea, but as soon as you find out, you go, oh my God, we've got to be careful with this stuff. And we saw that we explained the basic science behind it. So that's not a reason not to use it, but it is a reason that you have to have clinical trials. There's also galactose, which is one of the components, one of the disaccharides or monosaccharides of lactose. And that can be used to treat several of these disorders. Fucose is another one. And you can use fucose to actually improve the conditions of some of these patients. Maybe not all of them. But again, we have to know and understand some of the basic science. But originally on the basic science, it shouldn't have worked, because fucose was thought to be, it's not metabolized, very much, maybe a little contribution to fucosylation. But in turn, it turns out that it can be a major contribution if you have extra supplements of fucose. And so all of this stuff was unknown. Now we're beginning to understand it. And again, it takes you back to the basic science. And there's one other component that is being used, not only in CDG-related disorders, but even there are ideas that this may be important in rather common disorders. And that's the use of N-acetylglucosamine. That is getting some evidence that it may be helpful in the disease is slipping my mind at the moment, but it's I'll figure it out. But there's evidence that it is improving some of the symptoms. So, I think that these sugar supplements are not normally thought of as medicines. We do need to validate them, but it's important and it can be used, but you need to do it carefully. And if you want to know what carefully means, contact the consortium that we have for CDG. That will give you a better insight.
How long ago have you been looking into, or in general, have people been looking into the potential supplementation of these sugars for people with CDGs?
We started it with mannose back in about 1996. And do you want the story on that?
Sure.
Sure. So we had cells from a patient. We didn't know what the defect was, and we were using culture media, but we could see that there was a deficiency in glycosylation. And because most of these glycans are made from mannose, we just thought, well, maybe throw a little mannose in. Maybe that would help. Well, the glycosylation got better. And so we actually went to the FDA in the US and said, can we try mannose on ourselves? There was no literature at all on this. Can we try it on simple sugar? The FDA within 3 weeks responded and said yes. So we lined up all the people that we had in the lab, and over 2 weekends, we dosed ourselves with varying amounts of mannose, multiple rounds, and then, of course, we would tap our blood and watch the absorption, the clearance. So we had real data. And it was actually, I guess, just a couple of weeks after that, a couple of days after that, that I had a call from a physician in Germany who says, we have a patient who has some sort of glycosylation disorder. We don't know what it is. And we saw that you published this paper saying, well, maybe mannose could help. Do you have any idea how much mannose to give? How often? I said, funny you should call. Give this much, this often. And the patient got a lot better. And then we realized that, in fact, all the biochemical parameters and his growth and all the other problems had improved based on the mannose. And so that was our first clue back then that, this is something that deserves exploration. And then that led to fucose, and then to galactose, and now N-acetylglucosamine. So it was sort of another one of those, it's basic science, let's take a look, but there's a lot more to the story.
You said several different treatments are being developed. Could you elaborate a bit about those?
Galactose is being used for a disorder called PGM1, which is a deficiency in phosphoglucomutase 1, that's glucose 6-phosphate to glucose 1-phosphate. That deficiency has a series of symptoms, and there's evidence that that is improving the phenotype of these patients. There's very good evidence for that now. So that is about to enter clinical trials. Galactose can also be used for a galactosylation deficiency, and that is in this carrier of UDP galactose into the Golgi. There's evidence that giving some galactose can help. In a deficiency called LAD2, leukocyte adhesion deficiency type 2, that fucose can help some of those patients. And again, it was not thought that this would make any difference, but in fact, it does, because they usually get immense amount of infections. They can't have their white blood cells extravasate to fight the infection in the tissues. It stays in the circulation. And the reason it does is because there is a fucosylated glycan, which is necessary to allow the first step before the white cells get into the tissue. And that step is blocked. Once you provide fucose, within a couple days, the sugar chain is made and the white cells can go in and fight infections. So that's another one. N-acetylglucosamine is a potential therapy for a disorder called NGLY1 deficiency. And I have to say that it was a parent who figured this out. And the way it worked was this parent, very, very bright, very involved parents, noticed that, or thought that maybe there wasn't enough N-acetylglucosamine for many reasons. And so they decided to dose their son with it. He couldn't form tears. And you get these crusty things on his eyes, and at one point they were saying, how can we take care of our kid? And they would put eye drops in to moisten, the tears because he wasn't producing them. They gave him N-acetylglucosamine. The crust went away. He was starting to make a little bit of tears. They took him off because they came to me and said, what should we do? I said, well, sorry, but you got to take him off of that now. And the crust reappeared. Put him back. And at that time, the patient's grandmother came to be with the boy. And because the parents were going to be gone, the grandmother said, I have never seen his eyes look so good. She didn't know about N-acetylglucosamine. By the time the parents came back and he wasn't getting any dosing, it had gotten worse. And she says, I must be a terrible grandmother. He's getting worse right in front of my eyes. So the experiment was done, and now there are a number of children who have taken this. So that N-acetylglucosamine is going to enter into a clinical trial, again, with all the right kinds of measurements probably within the next year. So it's these sorts of things where, you're bootstrapping, you're again in this ecosystem, and everybody's making a contribution.
It's absolutely amazing just what understanding of basic science can get you. It's real detective work.
Yeah, it's a lot of fun, I gotta say.
Now, one thing I want us to consider before we conclude our conversation is funding. These are very rare conditions, and so could you comment on the challenges of finding research funding for these disorders? Sometimes
it can be very difficult. And we've been fortunate in getting funding from the NIH. And I think at least part of that was showing that there was a relationship to a disorder, but also bringing in the fundamental science. So the NIH loves fundamental science, especially when it applies to patients. Now, there probably is a break-even point where some reviewers, or maybe even some people in the NIH administration would say, well, that's just too rare. I hope that's not the case. And if the opportunity to make contributions and understand basic science and affect even a small number of patients can be done, that the right group of reviewers can look at that and say, this is really important. I have also, because of my familiarity with that system, realized that there are not all that many people trained in glycobiology, glycochemistry, who can appreciate that connection. So if those people are not on the review panels, they go, well, I don't think this stuff is very important. I mean, I study phosphorylation, or I study other things. I can't see that that's important. You need people on those review panels who know glycosylation. And not every glycosylation grant is going to be up at the top. You can stand improvement. But that's one of the things that we need to do, and that's where the education is really important. The other aspect, I have to say, and we've been extremely lucky in this, is that sometimes there are parents and families of patients that we've worked with, and sometimes they have been so incredibly generous to the lab, because they realize our connection, and they want to support us. And there have been literally hundreds of donations to the lab. Some from I don't need birthday presents redirected to our lab. Another person might hold a fundraiser in a bar, and then provide us with the money from that. Somebody ran the Grand Canyon as a marathon and sent the money from that. And there were others that were $10 and $20 donations. Some of my family might send something, and I donate. I mean, so it's, if your heart's in it, you're connected, that helps. But we need the big hundreds of thousands in education to take care of that.
And especially when it comes to scientific advocacy, could you tell me a bit about FASEB?
Yeah, FASEB. FASEB in the U.S. is an organization of over 30 biomedical research societies: genetics, physiology, glycobiology, biochemistry. And they have a special place in advocacy because we as scientists can go to Capitol Hill, sponsored by FASEB and organized. And the people that do this have credibility with the staff people and in Congress. And they can set up meetings for us, and we can go tell them the stories about what is going on, what's happening, and how that benefits their own district. We don't have to convince our local congressman. He knows that very well. He's visited the lab a couple times. And so they realize the economy of San Diego is influenced by it. But there are people everywhere who are impacted by disease. And we can bring in, sometimes we bring patients along with us. And I can guarantee you that's the thing that moves people the most, is when you walk in with the patient and you have them meet the staffer or the congressperson. That helps. And so that kind of thing. And stressing that idea that you don't get this unless you have the basic science. So taking care of basic science from Thermus aquaticus to N-acetylglucosamine is worthwhile, and it's going to help people in your district.
No, I find it crazy what direct contact can achieve.
Makes all the difference in the world.
I think a very good place for us to conclude is for me to ask you for advice for parents whose child might have been recently diagnosed with a congenital disorder of glycosylation.
Especially if you're in the U.S., contact CDG Care. They're a really great organization. And I think if it's internationally, then the World Conference is a good place to start, and you can have all of the connections you'll need there. They have resources for educating families. They have simple videos that educate parents and educate physicians, about what CDG is, and we have special areas for professionals as well. So contact that. And the other one, of course, is the Frontiers of Congenital Disorders of Glycosylation Consortium in the U.S. You get those on your list, reach out, and I can absolutely guarantee you're going to hear from people, probably sometimes within minutes, but it may take a day.
Thank you very much for this conversation. What you're doing is incredible. I think just that the journey you've taken from slime mold to the commitment you've had is really, really something to applaud. And thank you for your persistence in this field.
Well, thank you, Rena. I appreciate this opportunity to answer all the questions, and the topics you brought up are the very ones, if I had to go out and give a lecture, that's what I would talk about. And you've just set this up so perfectly. You're quite a good interviewer, and I very much appreciate it. Keep up the great work, okay?
Thank you very much. Now, speaking to our listeners, I hope this conversation helped you better appreciate the integral role of glycosylation in normal physiological function, as well as the importance of collaboration between scientists, clinicians, and patients. As Hudson said, we are all a part of the same ecosystem. If you would like to access more information about this conversation, Hudson's research, and links with useful materials for patients and professionals who want to learn more about CDG, follow the link in the description to the show notes for this episode. Equally, if you want to find out more about GlycanAge, head on to GlycanAge.com, where you can access a whole list of our scientific publications, blog posts, testimonials, and of course, this is where you can order your GlycanAge test kit. Watch out for our next episode, where I will be joined by Samia Mora, a cardiovascular health specialist whose research focuses on risk factors and prevention of cardiovascular diseases. We'll be discussing cardiovascular disease risk factors associated with lifestyle and biological sex, as well as the potential use of IgG glycans as prognostic tools. Please don't forget to leave ratings and reviews for this episode and engage with us on social media. Thank you for listening and have a great day.